SABCS, we are able to educate our patients. We're able to bring advocates to us and have them tell us what they want and what they need from us, but also is able to bring researchers together to start the conversations that we need to have for our new clinical trials.
I think the symposium helped to bring together all the people around the world that do breast cancer research, from Europe to Asia. And I think that's made a major impact. I have many friends now throughout the world because of this symposium. I think it also spawned some similar meetings now ongoing in Europe and other places to spread the word about breast cancer research and the accomplishments that we made. So I think it's had a major impact across the global spectrum.
I think that we also will be doing way more research on breast cancer health disparities. For the last several years, I think we have not paid enough attention to breast cancer health disparities, and I think there's going to be an increasing impetus and emphasis to focus on that.
I think that clinical trials are vital. I think that anything that I can do to help researchers further the work that they are doing is important.
As an advocate, my message to scientists and researchers is to continue to involve patients in early development of their clinical trials and to also include the diversity of these patients.
But just knowing that there is something that I can do, not only for myself and for my family, but for the broader breast cancer community is so rewarding. It has afforded me a seat at the table. But not just a seat at the table, to be in community at that table through sharing, which benefits all. It benefits the patients, the clinicians, and the researchers. And that has been so very important to me.
I mean, totally invaluable when they all come together and congregate and they share knowledge and they disseminate knowledge. I don't know how they could do it without these kinds of meetings. It was exceptional. There's nothing that can replace it, in my opinion.
A cure would be a wonderful thing. It would be something that would be unbelievable. But anybody that's metastatic, sure, we would love a cure. But what we really want is to stop progression. It's as good as gold for us.
Good morning, everybody. My name is Virginia Kaklamani, one of the co-directors of SABCS. Thank you for being here relatively bright and early, the last day of the meeting. Every time I see this video, it just reminds me of why we do what we do, and we do this for our advocates, for our patients, so I truly appreciate everybody's effort. So today's plenary talk is going to be by Professor Khaled. He's Professor of Tumor Initiation in the Department of Pharmacology and the co-leader of the Human Breast Cell Atlas Initiative at the University of Cambridge and the Wellcome MRC Cambridge Stem Cell Institute in Cambridge, England. Dr. Khaled obtained his PhD at the Department of Pathology at Cambridge with Professor Christine Watson, focusing on mammary gland development. He was one of the first to use RNA-seq to identify cellular damages in the normal and pre-neoplastic mouse and human mammary gland. And him and his team have done a lot of work, both mouse and human, identifying several pathways that are associated with precancerous cellular changes in women at high risk of breast cancer. This includes observations that immune cells in the healthy breast tissue from BRCA1 and 2 high-risk women exhibit an exhausted phenotype. His talk is going to be on cell atlases as roadmaps to cancer immunology and prevention. So I know it'll be inspiring for all of us. Dr. Khaled.
Okay, good morning, everyone. First of all, thank you very much for the invitation. It's an absolute pleasure to be here today to tell you about our work. And also, it's my first San Antonio, and congratulations to the organizers. It's been a fantastic meeting, so definitely coming back. It's a great event. So what I'm going to tell you about today is the work we've been doing for the last 8 to 10 years, focusing on using single-cell transcriptomics and building atlases to understand the early precancerous changes that occur in the breast, and hopefully exploit this information for preventative therapeutics. So these are my disclosures. So I was a bit nervous using this slide, but I'm going to go ahead anyway. So why therapeutic cancer prevention? I think it goes without saying. Cancers are quite complicated. There are a lot of issues. Of course, you guys, we've heard fantastic new advances in this meeting around clinical care. And I think it's the old saying goes, prevention is better than cure. But it's been quite challenging, of course, to do this. But I think bill payers and policymakers are really forcing us all to think more about this. And what I would like to really do in this talk is hopefully convince you that maybe we're at a point where we shouldn't be stopping doing what we're doing already, the great work, but maybe paying more attention and developing new ways of therapeutic prevention. So what is the current state of play? Well, this is a slide from a WHO report on early diagnosis of cancer, and it kind of oversimplifies tumor development, starting all the way from healthy cells to metastasis, and someone unfortunately dying from disease. Of course, you're all aware, most of the cases tend to present with symptoms, so they go see a doctor, and I think that's where we get treatment, and I guess that's kind of the traditional approach to treating cancer. The earlier you are in terms of in that process of picking up the disease, the diagnosis, the better are the chances, of course, and better outcomes. In breast, of course, we have screening programs, which we could have debates if they're good or bad with overdiagnosis, but it's not for today. But really what I would like to focus on is this bit here, which I think we're at the point now where we have a lot of information and accuracy in terms of identifying these abnormal changes, so asymptomatic abnormal changes in tissues that could and lead to tumor initiation later on. So in the breast, I think I'd like to give a shout out to a couple of papers which have really been quite inspirational for me at least. One from Jeff Lindeman, who's here, I think, and he's presenting later today, so it'd be great to see their updates. And from Matt Smalley, and I think what they've shown in these two papers that in the breast, at least at high-risk individuals, that there is certain populations that seem to be at higher risk of being the putative cells of origin for tumor development. But also it kind of highlights, which is, sparked my interest in this field, that there are potentially abnormal changes that could be targeted early on to prevent tumor development. So how do we tackle this in my lab? We have 3 big questions. What are the precancerous cellular changes that occur in a healthy tissue on its trajectory to tumor development? Can we detect these changes early enough, and can we prevent them from, or can we reverse them rather to prevent tumor development? You might have noticed I've used healthy between quotation marks because, of course, the breast is quite dynamic tissue. So it's a ductal epithelial structure embedded within fat stromal immune cells. And it changes quite dramatically, of course, in response to gestation, lactation, and so on and so forth. And what's always fascinated me since PhD time is that some of these normal developmental changes are also risk modulators, right? So they modulate the risk. So we know parity, protective, age is a risk. But up to this moment, really, we haven't really had detailed cellular mapping, if you would like, of how the tissue changes in response to these changes. So we set out to build this map. So we need to draw a map of how the breast changes in response to normal healthy conditions. So almost 8 years to the day, we did our first single-cell RNA sequencing experiment where we basically isolated epithelial cells from the mouse from 4 different developmental stages, and then performed 10x single-cell genomics. So we used nulliparous mice, gestation, and lactation, and post-involution. And this was a simple experiment by nowadays standards, where we just looked at the epithelial cells. And to cut a long story short, that basically showed us that we could capture the different states of the epithelial cells within the tissue. And there are about 15 different epithelial subtypes that you can gather from this data. And this tend to represent either cell types that are found during normal certain developmental stages, and even cell types that maybe have memory of having gone through certain developmental stages, such as gestation. For those of you who have not seen single-cell data, which I'm sure you all have, each dot on this plot is a cell, and basically we're looking at here even a more simplified version of it, where we're kind of summing down the expression of the 20,000 genes in that cell, and cells that have similar expression are clustered together, so they form a cluster, and then now we color them in this pseudo-colors to show that they are similar. So I also need to say, around the same time of our paper, Jane Visveda's lab also published a very similar study and very similar outcomes as well. Okay, so with this reference map in hand, we then set our target or our aim at a much more difficult question, which is, could you then use this technology to look at what happens in the tissue during precancer? And we did this using a BRCA1 mouse model. So this takes about a year to develop tumors. And we took mice 15 different time points, starting from about 6 months of age all the way to tumor. So we had 2 tumors in the study. And what we did here was isolate. So unlike the first study where we focused on epithelia, we decided actually it would be really interesting to see what happens to the entire tissue during these precancerous changes. So we isolated, broke down the tissue into single cells, and basically took a sample and sequenced a representative of what's happening, so including immune, stroma, and fibroblasts. Unfortunately, we can't recover adipocytes, which is a drawback to this approach, but there are other ways of doing it now with spatial technology. So this is all published, so to cut a long story short, basically we could now capture a lot more cell types, so about 53 different cell types that you could capture that way. So for simplicity, we grouped those 15 time points into 4 different precancer stages, 1 to 4, and I apologize to clinicians. I realized using the word stage is not a good idea, 1 to 4, but precancer 1 to 4, and then also tumor. And really, just to cut through the detail, but anything that's red or orange is basically an immune cell compartment. Anything that's green is basically a fibroblast. Anything that's blue is epithelial, and the tumor you could see there in purple. And you could see, I think, during these precancerous stages, that the tissue changes quite dramatically, and these compartments change quite dramatically. You could look at it in a bit more fancy way. So we could animate this UMAP that I showed you earlier. So here again, same concept. The blue compartments are epithelial, the red, orange are immune, and the green are fibroblasts. I've highlighted a few compartments that are worth noting. So you could see the cell of origin that I talked about earlier, the luminal progenitor. They change and shift to the left. I'll come back to that in a second. But if you even ignore the epithelia, which of course are of interest, you could see that there are a lot of changes happening in the immune and compartment before any tumors appear, and in fibroblast compartment equally as well. So we see, if you take a kind of a statistical agnostic approach and ask the question, which clusters go up during these precancerous changes and which ones go down, you see there's actually a majority of them are immune cells. So you get a lot of increase in a lot of the immune compartment, and obviously that's you can almost see it by eye as well. But what's interesting is they all seem to be tumor promoting. So the CD8 cells are even exhausted. We have an increase in Tregs and macrophages that are tumor promoting. There's only one epithelial compartment that we found in this study that went up during this precancer, and this is this AVD, which stands for alveolar differentiated cells, and this was a big surprise to us. So these are the milk-producing cells, right? So they're normally only found in the mouse during gestation and lactation. These mice were nulliparous, and actually they are the direct precursors of these luminal progenitors, the ER-negative luminal progenitors. So it seems to be, now a few years on, we now know that these luminal progenitors undergo an aberrant differentiation, probably in response to the DNA damage that they are carrying towards this alveolar differentiated. We've seen now the same thing in BRCA2 mice models, BRCA and PALAP2 as well. But it's really nice to see that the community has also started to report this. So other types of mammary tumor development or mammary models, tumor models are showing a very similar observation. So this is not all just computational RNA-seq. We could see these changes at the protein phenotype level. At the epigenetic level, these cells become hardwired differently towards kind of an epigenetic state of differentiation. And actually, this was the first time we spotted some of these changes also in some of the human samples, which I'm going to focus the rest of my talk on. So of course, what about the human breast? This is all mouse. I think a lot of people in the room say, oh, who cares about mice? We're not going to cure mice. We want to know about human. So we started the Human Breast Cell Atlas. This is in 2019. So this is a huge team effort between my group and John Marioni's group, and which I'm going to talk about for the rest of this lecture. I'm not going to mention the first part, actually, of this, which we published, which was done by a talented postdoc in the lab, Alicia Jane Twigger, where she actually sequenced cells found in human milk. So there are live cells in human milk, which was a big surprise to me. And it's fascinating, actually, this study, and I encourage you to read it. Alicia's now an independent group leader herself in Cambridge, and she's setting up her lab. You should look her up. She's doing great stuff. Okay, so the rest of the atlas was basically just published earlier this year. And this basically was an effort where we got samples from a tissue bank called Breast Cancer Now Tissue Bank. And it's a great collection that they've got. These are normal or samples that are not cancerous from individuals that they also have a lot of metadata on. And I think this is really important. I think any sequencing study that doesn't have metadata is basically meaningless. It's rubbish in, rubbish out. So this was critical for us to have this data so that we can make the correlations and the important observations that I'll show you. So we had samples from 55 donors, 22 reduction mammoplasties, 11 BRCA1 prophylactic mastectomies, 11 BRCA2, and also some samples from other women that have family history of breast cancer, but not BRCA1, BRCA2 carriers, and some contralateral mastectomies. So what did we do with the samples? Sarah Pencer and Jack Stenning did a tremendous effort. Obviously, we wanted to standardize everything and make sure this is done properly. So they isolated 3 different compartments from each sample, stromal immune enriched, epithelial enriched, and we only fact sorted for luminal progenitors. And these are, as I mentioned, the cell of origin or putative cells of origin. This was all done during COVID. So Jack filmed himself on a GoPro doing this. So there are 10 videos on my lab website if you ever want to do single cell dissociation of the mammary of the human breast for sequencing. You should follow Jack. He's walking you through it. And it's actually a nice resource to make sure that it's reproducible. So this led to 161 samples and 1 million cells sequenced, which is the largest collection out there at the moment. So this then was handed over to a very talented mathematician, PhD student in the lab, Austin Reed analyzed this data. And I'll walk you through this analysis now. So here are the 3 major compartments of cells that you find in the human breast. So epithelial cells, so we capture different flavors of ER negative, ER positive, and basal cells. We see a lot of stromal fibroblasts as well, and lymphatic endothelial cells, and the different flavors of immune cells. So now we have this data. What do you do with it? It's obviously quite a challenge when you have humans are not mice, they're different. So how do you start asking questions? What goes up with age parity? So actually this is where collaboration with John is really important. So at that time, they developed a computational tool called Milo, which I don't know the math behind it, so don't ask me. But basically, this was done with Sarah Teichmann's lab as well, which allows you to ignore these clusters labeling and actually at the neighborhood, small neighborhood level, start asking questions within a few neighborhoods, how many cells are enriched with any condition that you're interested in. So it allows you to kind of gain statistics at great levels. It's computationally quite heavy. So when you do this, so now I'm kind of coloring the same plots that we have on the left, but now with this Milo analysis. So anything that lights up blue means it's an epithelial, is a compartment that has been enriched with whatever question we're asking. So at the top here, I'm showing you things that are enriched with age. So you could see, for example, there is a hormone ER-positive population that goes up in age samples. And interestingly, these are the same population that seems to go down in samples that have undergone parity. The full data set is obviously in the paper. You can have a look at more of the other changes. So now to the BRCA1, BRCA2 samples. Of course, they are of interest. Similar results here on the left. But we've here summarized it also in the plot on the right, which I think is a really nice way of looking at tissue state. So on the left, we have the different cell types here on the y-axis. And the zero line is effectively what an average risk breast would look like. And then we plotted how the BRCA1, BRCA2 deviates from the average risk. So effectively, you could then plot that. The BRCA1 is the dark blue line, and the BRCA2 is the light blue line. So effectively, you've got like a profile, fingerprint profile, if you would like, of what these tissues would look like and how they deviate from the average risk. Two things to note. There's a lot of changes, of course, but two things to note. Epithelial changes seem to be different between BRCA1 and BRCA2 during precancer, which might account to some of the clinical differences, I guess, in types of tumors that you would get. But there are some changes that are common. So this was here, the immune compartment, very similar to what we saw in the mouse. There was an enrichment in these immune cells coming in precancerous tissue, right? So a lot of CD4, CD8 cells. So we validated this orthogonally. So here we used the multiplex immunohistochemistry, a panel of 8 antibodies for, again, CD3, CD4, CD8, CD68, FOXP3, PD1, PDL1, and pan-cytokeratin to mark epithelia. And luckily, we had samples and slides from FFP samples and slides from the same women that we sequenced in the atlas. So 30 of these samples were also stained. So it's kind of, you could match the data, which is great. And it gives you obviously very, very, very pretty images. But importantly, it validated our finding, which was a great relief. So we could definitely see these immune infiltrates happening in the BRCA1, BRCA2. We could actually quantify it. But what was really interesting and surprising at this point is there seems to be this immune exhaustion phenotype. So here, these are PD1, CD3 cells, and you can see there's an increase in both BRCA1 and BRCA2 compared to average risk individuals. So this immune exhaustion happening in effectively normal tissue before any tumors arising. And this is not confined only to to PD1. Other ICBs as well, immune checkpoints are also upregulated. We see an upregulation of the ligands in the epithelial compartments. So the ER-negative luminal progenitors have high levels of PD-L1, so are the hormone sensing, and even macrophages as well, suggesting that the entire microenvironment is effectively conditioning the tissue towards this tumor initiation to occur. So I mentioned we have a lot of cells per individual, so we sequenced a lot more than other efforts. So have we got any evidence in this large atlas of tumor initiation? Could we see anything abnormal? So those of you who have keen eyes might have noticed in the epithelial compartment, there were these two clusters that are labeled DDC1 and DDC2. So these stand for donor-derived clusters. And the reason why we call them that is that they came from two different donors. They had profiles that did not match the traditional clusters of epithelial cells. And you might have also remembered I said that we were very conservative with quality control. So although we've sequenced 1 million cells, we got rid of 200,000 because we didn't want to put out an atlas with rubbish in it. So we got rid of doublets, dead cells, and debris. So we're very conservative with our quality control. And these DDCs survived quality control. So they're not, we think they are not artifacts. So we decided to look at them in more detail. And first thing we did is see what they express. So here are a bunch of markers that normally delineate the different clusters of epithelial cells in the gland, so luminal and basal flavors of the epithelial cells. And you could see that normally basals and luminals don't mix in terms of their markers, but actually DDC1s and DDC2 had a mixed lineage phenotype, which is, of course, we know tends to suggest it's the early steps of tumor initiation. So we only had RNA sequencing, of course. We didn't have any DNA sequencing to look at, but you could computationally infer copy number variations using the single-cell RNA sequencing. So let me walk you through this plot. So here each row is a cell, and each column is obviously a chromosome. Anything that's marked as red is a copy number gain. Anything is blue is a copy number loss, or predicted gain and loss. So here we've got samples from DDC2 and samples from DDC1, and you could already see clearly that there's a copy number gains at 1Q. We could also see copy number gains at other regions of interest. So here, for example, this is cyclin D1, predicted cyclin D1 region, and this DDC2 seems to have a putative copy number gain at HER2. So this was all very interesting. And of course, because these clusters were sitting out from the normal clusters, we asked the question, because we have the rest of the cells from the same donors, could we see any compartments within the normal epithelia that would suggest that they were the cell of origin, right? Does that make sense? I hope so. So we did the same analysis on the rest of the cells from DDC1, who's in this case actually is a BRCA2 carrier. And it turns out there was clearly a subpopulation of the subpopulation of luminoprogenitors that have this copy number gain at 1Q, also other copy number gains that are conserved, suggesting that it's maybe a subclone of these cells really gave rise to these putative tumor initiating events. Similarly with DDC2, same thing. So this HER2, we spotted also in luminoprogenitors a small population, so a clone within the normal compartment that seems to suggest that there's a relationship between these two cells. So I think that was really quite interesting. We did the same analysis on the entire cohort of the atlas. Please don't post this because it's not published yet. This was really quite interesting. So here the blue line is average risk individuals. And you could see just in the normal cells, as we get older, there's an accumulation of copy number variations, right? So that seems to go up as a factor of time, which is obviously interesting as we are all getting older. The green line is actually BRCA1, so there seems to be an accelerated aging phenotype, if you would like, happening, so increasing copy number gains, which I guess makes sense. What was really surprising to me, and I think we should take it with a pinch of salt because we need bigger numbers, is BRCA2 doesn't seem to have the same effect on the tissue. But I think this is all preliminary, and we need to do more samples to be able to gain this information. Mouse actually does the same thing. So old mice have also increased copy number gains in normal tissue. And the red line here is BRCA1. You could see the gold, green, brown line. I don't know what color that is BRCA1 with a knockout of gene X. We know that if you knock out this gene, we can actually prevent tumor development in BRCA1, then actually that seems to kind of abrogate some of these copy number effects. Okay, so hopefully I convinced you that doing these kind of atlases is useful. We've put all our data out there, so if you take out your phone and scan this QR code, it will take you to this website called Cell by Gene, which is a fantastic resource funded by the Shan Zuckerberg Initiative. So we've put all our data there. It's a user-friendly website. You can put your favorite genes and look and see where they're expressed in these different compartments within the human atlas. You can also color the compartments based on some of the, the different cell types, so level 2 annotation, here we call it. But also we have some metadata that we've uploaded on there. Not everything, unfortunately, but some of the metadata. So for example, BMI is super interesting. I'd love to study this more if we had more samples. But you can see some clusters are clearly enriched with certain higher levels of BMI, similarly for age as well. But since this is a service to the community and as part of the Human Cell Atlas, so Austin did a heroic effort. So although our atlas is the largest out there, we combined it with the other great efforts that people have also published and made an integrated human breast cell atlas. So this is part of the paper. You can see it, and actually is going to be version 1 of the breast atlas as part of the Human Cell Atlas. So this is now 2.1 million cells from 286 individuals. And I hope this is a resource that the community can use for their studies. Okay, so what have we learned so far, and what are the next steps to this kind of studies that we're doing? So obviously the cellular composition of the healthy breast is quite dynamic. I hope that's obvious and clear. Healthy breasts do have subpopulations of cells with aberrant copy number variations. which is, I guess, very similar to other tissues have reported mutations as well, that we're all walking around with mutations. BRCA1, BRCA2 tissues exhibit a pre-cancer immune exhaustion. I think that's something we are following up as a way of kind of intercepting and hopefully to see if that could be used for prevention. I know clinicians look at this and say, you know, this is mad because of given the toxicities of these type of drugs, but we're trying to see if we could play around with the conditions and how we could use these to make them safe. One message that I hope you could all take away, I think we should all be thinking about the tissue state rather than a single biomarker or a handful of biomarkers. How does the entire tissue actually respond is really quite important. I think what we should be looking for is biomarkers of waning resilience. You know, since we're all walking around with mutations, there must be some hardwired resilience mechanisms within tissue, not just immune cells doing the monitoring, but actually structural and other types of tissue resilience. And speaking of tissue state, the mouse gets quite a bad rep. Actually, if you look at the tissue state of the BRCA1 mouse compared to the BRCA1 human tissue, you could see that now we could see very similar, not exactly identical, but very similar kind of tissue state response to having a mutation such as BRCA1. And now we're actually exploiting this fact to try some preclinical trials that are ongoing in the lab, which we will hopefully try and see if we could prevent these precancerous changes in the mouse model as a kind of precursor before going to human studies. We're testing few therapeutics, and these are all approved or near approved drugs, so hopefully with making window of opportunities easier to go to quickly. And we're also developing ways to measure these early precancerous changes in a non-invasive way. Final point, which is something we all need to be thinking about if we're going down the prevention road, is how we're going to measure success in a timely way. No one wants to wait for 10, 15 years to see if tumors will develop. So we need to think of different ways of doing it. We also need to develop different types of drugs. We can't be developing the traditional oncology approach of drugs that kill cells. So I'm going to stop there, thank the team and my collaborators throughout the years. Big shout out to John Marioni. It's been fantastic to work with him over the years. He's now at Genentech, so he's a great loss to Cambridge community. All the data sets that we generate are on my website. We have kind of user-friendly websites that you could mine the data, but also you can download them, of course, and reanalyze them. And the guys at the bottom pay the bills, which is super important. Thank you very much.
Thank you, Dr. Khaled. This was an exceptional talk. So we are going to follow with general session 3.